Muchas gracias, Stuart. Y ahora le toca el turno a Nicoletta Landsberger. Nicoletta Landsberger es la directora del Instituto Científico de San Rafael en Milán, Italia. El Centro de Investigación de San Rafael se estableció en el año 2011 debido a la voluntad de la Asociación Italiana de Padres ProRed, y desde entonces este centro es altamente apoyado por la investigación ProRed y es dirigido por la doctora Nicoletta Landsberger. Toda la actividad de investigación del grupo se dedica al síndrome de Rett y a los trastornos relacionados con el MeCP2. En particular, están desarrollando y caracterizando modelos de ratón noveles de MeCP2, identificando nuevos genes o vías de MeCP2 que podrían ser relevantes para síntomas y tratamientos específicos. Y también están traduciendo nuevos resultados o hipótesis en estudios terapéuticos prospectivos para el síndrome de Rett. Cuando quieras, Nicoletta.
Yeah, so I have to stay closer. Okay, so I would like to thank the organizer for giving me the opportunity to present some of our work today here, and I want to apologize with all the families. I really didn't realize this was supposed to be a talk mainly for families, so I prepared a talk that was completely scientific for scientists. So this morning I tried to arrange it, but now the topics was already decided. So I will try to make any effort to explain why we think it might be important. This research that I am presenting, that is not the only one that we're doing in the lab. So what I was thinking of doing is to give you a brief introduction on MeCP2 function, although Stuart has already done it, and the importance of using mouse models, and then to show you just a couple of slides on some already published results from our labs that I think testifies first of all, that MeCP2, as Stuart mentioned, has a role during embryogenesis, and also that maybe by studying MeCP2 function early during development, we might find good deregulated pathways that might be a target for therapies. And eventually I will go on the most tough part of the talk, that is going to be about this novel mouse model that we generated, and I will try to explain you why we generated it and why we think it is important to generate novel mice, what they can still teach us. So as Stuart told you, MeCP2 was identified originally by the Adrenberg laboratory, and when he identified it, he defined this as a protein that can bind to DNA because it's methylated, and then repress gene transcription. So it represses gene expression. However, along the years, several other functions have been found and described for MeCP2. So it has been proposed always by Adrenberg in 2010 that only in mature neurons, so meaning when the brain is already adult and in neurons, the level of these proteins are so high in abundance that now the protein starts to work as we call it an architectural factor. So it means that the protein starts to organize the DNA in our cells in order to be very compact and in order to properly control the genes that do not have to be expressed are indeed silenced. Okay. Someone else has proposed that the protein might have an activator function, might be important for regulating the maturation of RNA molecules. Our laboratory proved that either directly or indirectly is involved in protein synthesis. And more recently, we demonstrated that the protein is associated with a particular cellular organelle that is called centrosome that has a role in cell proliferation. So from all of this, you really can understand that this is a versatile protein in terms of function, and we still don't know, although Stuart hypothesized only one function, which one are really relevant for Rett syndrome or other diseases, because you need to keep in mind that MCP2 is involved in several different neurological diseases, and is a cause of a neurological disease for one human being every 4,000, so really a lot of diseases. Not only Rett syndrome. Rett syndrome is the tip of the iceberg, okay? So what we don't know is exactly which are the real important functions, and the other things that we don't know, I think, is MSCP2 performing all this activity along brain development and in adult, or is maybe specialized to do something in a particular time window and does something different in another time window. And this is important for therapies as well, because we have to understand when to cure, when we are curing, what kind of molecules then to use. However, oh no, let me say one more thing. However, I do think that if we analyze all the function of MCP2 that have been identified so far, apart from the centrosome, the bottom line is that the role of MCP2 is to regulate gene expression. So this, I think, it is the main functions, and I think that when we have to look for therapies for the disease diverse from gene therapy, we have to find pathways, molecular pathways that are deregulated in the absence of MCP2, and that can be druggable. So how to find these pathways? So to find these pathways, we need mice, and also to understand MSCP2 functions. So what kind of mice do we have available so far? The two mice that have been mainly used so far are the so-called MSCP2 null mice. What does it mean? It means that there is no gene. We are studying only the males, and because of that, all the cells of the body are lacking the protein. But however, you want to keep in mind that the disease is generally occurring in females, that therefore will have one normal gene and one gene mutated. So half of the brain will actually express a normal protein. And the other thing you want to keep in mind that usually patients don't lack the whole gene. They have what we call a missense mutation, or a mutation that leads to a shorter protein, but they don't lack all the protein. So we are not completely confident that the MSCP2 null mice, and this is the message I want to convey, is the best model to be used. We think we should run parallel studies with different mouse models. The other models that we have available are the one that Stuart also introduced, that are so-called the conditional mice. What does it mean? It means that we can remove MSCP2 either from selective tissue, the cerebellum, the frontal cortex, or the skeletal muscle, to understand what is the role of the protein in a specific area. Or we can remove MSCP2 in a different time windows, only in adult, in puberty, or whatever, in order to understand when it is important, okay? But now recently, I think most of the scientific community start to understand that it's important also to have the so-called knocking mice. Knocking mice means that they have only a single mutation. So generally what happens is that either they have a missense mutation, so an amino acid has been modified, like the T158M mutation, the most common mutation in patients, or they might be modified in a different residue, but generally they want to mimic human pathogenic mutation. Okay, all these mice, as Stuart have described it to you, suffer of similar features that can be more or less severe, depending on the mutation that we have introduced, and that pretty well mimic the human conditions. So the males have a reduced lifespan, but not the females. They're suffering what we call clasping. That is an indication of a neurological problem. It means that they close their backwards legs when we pull them from the tail. They suffer of breathing abnormalities, mobility impairment, cognitive defects, epilepsy, and they have reduced brain size. So they really well mimics the Rett syndrome. And all these symptoms appear later, is a late onset, as it is the disease. So this is why we believe that this is a good model for studying Rett syndrome, okay? But the animals so far have been interrogated, have been used mainly in adults in the so-called symptomatic phase or when the disease were already overt but not so severe. And the reason why they have been used in this period is because there were two assumptions. The first one, as you can see from this review from Huda Zobbi, the first one was that MeCP2 is not present in the embryonic brain, or it is very, very low in abundance. And the second assumption was that there was not playing any major effect because the disease onset is after birth. Okay. Actually, what we did in our laboratory is very much interested in finding what can be the role of MeCP2 during developing of the brain and during embryogenesis. And we think that this might be important to really understand its primary effect. So what we did in this paper that we have already published is to study what happens during embryogenesis. And what we found is that any single cells of the brain, any neurons, even neural stem cells, so cells that are still staminal, are expressing MeCP2, although they are lower in abundance with respect to mature neurons, as Adrian Burr suggested. And if we go to look if there is any defect during embryogenesis, the response is positive. So the slide that you can see here says that there are defects in gene expression and that these defects can be the cause of a functional defect. And in fact, we found that during embryogenesis, the neurons are less capable to respond to stimuli because of the lack of MeCP2. So this has been performed on null animals. Okay. So these results, we found this, the neurons during embryogenesis are less mature, are less able to respond to stimuli, and these two different phenotypes might be interconnected because it is very well known from neurologists that activity promotes maturation and maturation promotes neuronal activity. So we started to think from this data that maybe we can try to increase just a little bit the activity of this neuron. So we are performing this experiment right now only in vitro and in vivo with a genetic trick. So what we're doing is just to boost a little bit the activity of this neuron with drugs or with chemicals and see if we can rescue the morphology and the activity of these neurons. And then we have only preliminary results. I'm not going to go into the details of this, but the response seems to be positive. So the neurons are indeed maturing much better, and right now we are checking the transcription has been restored, and we're checking right now if they are also functional. So if this will be the case, it might suggest that in fact, drugs that boost a little bit the activity taken in young patients might be effective, but only maybe in young patients. So this is something that we also want to address. So with other study that we published, and again, this is too technical, so don't look at it, but I will just convey you the bottom line message, was we demonstrated that MeCP2 activity change along brain development. So it is very true, the protein is very tightly bound to this DNA that is packed into the nucleus that we call chromatin when the brain is mature. But instead is not so tightly bound when the brain is not mature. So like in the perinatal period, what we can see is that the protein probably binds and goes off, whereas in the mature brain is tightly bound, so it's changing its activity, okay? So considering this data, let me go, skip this. What we decided to do is what in our lab we are trying to do is to identify deregulated molecular pathways that can be druggable, okay? But we decided that we need to really think which could be the best model to use, the best animal. So to perform these studies, we decided that we would like to use two different mouse models. One is the conventional, the null animals that we all know, but another one for us should have been a knocking mouse, so a mouse that was mimicking a human mutation. And also we decided that it would have been important to study the results at different time points, so during embryogenesis, in the perinatal period, when this disease starts to be really evident, and in the adult brain to understand how the different features at the molecular and behavioral level changes upon time. And let me also say that the difference between having a null animal or a knocking animal is that if you don't have a protein that is very much important, the system will compensate. What does it mean? It will find molecular tricks to survive, although a really important protein is missing, okay? So, and in fact, Landsberger demonstrated that this trick exists, as you will see soon. On the contrary, if you have a knocking mutation, might be that the system does not feel the necessity to compensate because the protein is there, it's just not functioning in the proper way. And therefore, either there are no compensation mechanisms or they might be different. Therefore, what I want to say is that we believe that it is possible, and this is what I want to show, that null animals and knocking animals are different at the molecular level, okay? And we have to keep this in mind for studying Rett syndrome. So keeping this in mind, several years ago, we still didn't publish it. We generated a knocking mouse model that is, because at that time there were almost no mouse available. This mouse has a human mutation that is in this tyrosine 120 that is substituted with an aspartate, but this is not important for you. There is only one patient described so far with this mutation. We got interested in it because of other studies on MeCP2 phosphorylation, so we don't have time for going into these details. But we thought it might be interesting to have these mouse models also because this residue is inside the domain that Stuart described you as important for binding to methylated DNA and is highly conserved in evolution, suggesting a relevant importance. So we produced the animals. It doesn't work, okay? The animals carry the mutation. We made sure that it doesn't affect the MeCP2 gene transcription so that the construct is valid, and then we went ahead characterizing the animal. And to our surprise, because we didn't expect this, we found out that these animals are incredibly affected. So these animals have, if we look at the males animals, they have a really premature death that is even before the mouse that has been described as reproducing the most common human mutation, that is the T158 mutation. So they die quite early, just a little bit later than the null animals. They start to accumulate all the symptoms that Stuart introduced, tremor, breathing disturbance, motor impairment, and so on. So they really well reproduce the disease. The females, the females are characterized by not having a premature death, but still they do show phenotypes, as it is the case for the Landsberger model. And these animals, as well as most of the mouse model of MeCP2, also reproduce the reduction in size or in weight of the brain that is also found in patients and is not given by neurodegeneration, but simply by cell shrinkage. Okay, so it was looking like this was indeed a good mouse model. We wanted to also characterize it a little bit better. No time to go into the details, but let me just tell you that it has really coordination problem, gait problem, problems in strength in the forelimb. It has cognitive problems that are assessed with working and associative memory tests. So it has more or less all the symptoms that has been described for the null animals apart from anxiety. It doesn't suffer of anxiety. And as all the other animals, it seems that these features onset later, I mean, not exactly in the not soon after birth. Okay, so this is a good model, but what happens in terms, but is exactly similar to the null animals. So why we find it peculiar? So what we tried to do is to try to understand how come just a single modification, just a residue that has been modified is so detrimental for the brain function. So the first thing we did is to try to understand if MeCP2 levels were, the protein levels were maintained into the brain. And we did this at different developmental timing. So as maybe the diagram underneath, you can understand from that one, this diagram shows that if we look at the level, if we compare the level of MeCP2 with respect to the level of the knocking protein, in embryogenesis is normal. in the perinatal period, so at P6 is normal. But now when the brain starts to mature, instead the level of our knock-in protein drops and it goes to 50%. And we can find exactly the same data simply by cultivating the neurons from this mouse in vitro. So we start to see a phenotype that is a reduction in the level of the protein, but only when the neurons are mature. But this reduction in the level of the protein do not justify the serious phenotypes that I have described, because it is very well known that in mice, a reduction of 50% of the level of the protein does not lead to such severe effects. So there might be something more. So we went ahead and we thought, well, let's see if it impairs, this mutation impairs the capability of the protein to bind to DNA, because it is in the region that is relevant for binding to DNA. So this slide shows you, but I give you only the result. The results obtained simply in vitro using a small portion of the protein, just the domain of the protein that is essential for binding to DNA. And what these slides tell us is that, in fact, the mutation is leading to an important impairment to binding of the capability of this region of the protein to bind to methylated DNA. Fine. But the question is this occurring also in vivo? Because it is very well known that MeCP2 binds to DNA because of its methyl binding domain, but also because of its C-terminal domain. So to answer to that, we went ahead and tried to compare the capability of the protein to accumulate on these dots that you can see on the left part. So they are called heterochromatic foci. So if you see the most left panel in div 15, for example, you see these dots, and you can see that the wild-type protein that is in the center of the panels is able to accumulate on these dots. So these slides tell us that the wild-type protein, in the beginning, when the neurons are not mature, is not accumulating on these dots, so it's not able to accumulate on heterochromatin, or maybe it's binding and detaching, as we have previously demonstrated. But when the neurons get more mature, instead it's tightly bound. And we can picture this simply with this experiment. If we now repeat the same experiment with our knock-in animals, these are cultured neurons, now what we see is that our protein is never able to accumulate on these dots. In other words, the proteins look like the wild-type protein when the neurons are immature, because none of the proteins are on these dots, but then instead, the two systems differentiate. So the knock-in animals are different in mature neurons, but not in immature neurons. So we wanted to confirm this with a biochemical assay using brain from animals, and the bottom line of these two experiments that I'm showing here is exactly what I told you. So what the normal protein, normal MeCP2 does is that in the immature brain is probably going back and forth out of chromatin or DNA, but when the brain gets mature, the protein is stably associated with chromatin. On the contrary, our protein is always going back and forth from DNA, is never capable to acquire this stability on chromatin. So then we proceeded and said, okay, so what does it mean in terms of at the molecular level? So we started to go back to the experiment and the model that Adrian Bird proposed in 2010. What Adrian Bird said is that in the wild-type upper panel, that is the normal situation, in the mature neuron, MeCP2 works as a condensing factor. So there is a lot of protein and the DNA is condensed. In the knockout animal, that is the lowest panel, what happens is that a compensation mechanism occurs. A different protein that is indicated in green and is called H1 substitute to MeCP2s. Because MeCP2 is lacking, the system expresses more of this protein, and this protein compensates. Somehow organize a chromatin structure that is compatible with life. Okay? The question is what happens in our system, because in our system we have the protein, it goes back and forth, so will the system compensate or not? Or will this affect the capability, as Stuart said, of bringing these co-repressor molecules on chromatin that are really important for the disease? So what we did is to address if, in fact, we could find a defect in recruiting these complexes on chromatin when we have the knock-in. We performed this experiment in vitro. This is the slide. I don't go into the details because it's a sort of complicated. We are exploiting a construct that has been prepared by Adrian Bird. It's a protein that is called a subunit of the anchor complex. This protein is able to go onto heterochromatin and form these dots only if MeCP2 is present. In these slides, we can show that, in fact, when MeCP2 is lacking, the protein does not go on heterochromatin. It does not go. It goes if there is the wild-type protein. It does not go with the pathogenic human mutation, with the knock-in mutation that we have generated. These, however, are performed in vitro. What happens in vivo? To respond to this, we use a different co-repressor complex that Huda-Zogby demonstrated that is also important for the disease. It's called ATRX. Huda-Zogby demonstrated that ATRX, again, goes on these dots of heterochromatin only if in the mature brain, only if MeCP2 is present. So what we did is to take the brain of the wild type and the knock-in animals and check where ATRX was. And the answer is that ATRX is where it is supposed to be, in the wild-type brain, but is not there anymore with our knock-in mouse, suggesting that, again, we have lost the capability of this protein to recruit on heterochromatin, on chromatin, the complexes that are important for the disease. So what happens, and there are the last 3 slides. So what is the functional effect of all of this? We think that the functional effect is that generally now the chromatin structure, instead of being really condensed, is a little bit more open, okay? So to check if this would be the case, we used 2 different assays. One is here, is this one. For the scientists, it's called microcochlear nucleus accessibility, but it's not important for you. It's just that we use an enzyme to probe how much is open the chromatin structure. And what we did first is to check the wild type with respect to the null animal, the bird mouse. And we found out, as I think it was already published, but I'm not unable to find the publication. I have a memory of it. But anyway, the null animal has no clear defect in this test. So the null animal somehow has well compensated, and therefore the structure seems well organized. On the contrary, if we look at the chromatin of our mice, now we see a subtle but consistent global opening of the chromatin structure. Okay. The second test that we use to prove the same thing is to look at the level of this 5-hydroxymethylcytosine. We call it the 6th base of DNA. Is a base that is in our brain, might have important relevance for gene expression, but is generally used now as a marker of chromatin accessibility. The more chromatin is open, the more you have of this molecule, okay? So what we did is to use an immunological assay to test how much hydroxymethylcytosine is present in the knockout and knock-in brain with respect to wild type. And to our surprise, we found that the knockout animals have a statistically significant decrease in 5-hydroxymethylcytosine, suggesting that the chromatin is actually a little bit more condensed. And this might be the reason why we never found the upregulation of genes that were supposed to be found. On the contrary, our animal does not have the knock-in, does not have this decrease, but actually have a non-statistically significant, but a tendency of having more of this base, suggesting that the chromatin structure is not more closed, but a little bit more open. So let me just conclude saying that although all these studies for the time being don't lead to any druggable molecular pathway, we think they demonstrate that the MeCP2 null, or better, they demonstrate that we might have different mouse model. They all look alike in terms of general feature, clasping, mobility, hypotonia, epilepsy, and so on, but they might be different at the molecular level. And therefore, we start to think that for understanding the disease, and who knows, maybe even for the patients, we have a little bit to stratify patients. And I like his model. I think we should use null animals, animals that have the incapability, have a mutation that is affecting the capability of MeCP2 to bind to DNA, And the animals that have a mutation that is increasing the recruitment of these co-repressor complexes on DNA. They are all affected, but at the molecular level, they are very different. And the question is now, are they the molecular pathways that are deregulated are the same one or they are different? Because if they are the same one, we can use the same therapies for any patients. If they are different, it is possible. The therapies might be personalized depending, of course this is a sort of a challenging idea, but I start to believe that maybe not all the patients are similar and that maybe not all the patients have to have the same, exactly the same drug. If we are designing drugs in terms of the molecular deregulation, not of course if we are performing gene therapy or stuff like that, okay? So, and I think I can stop here, but let me just thank the person. So, since I'm running out of time, I'm not thanking the colleagues and the collaborators of the lab, because this is not really relevant for you today, but I think it's important for me instead to thank one association of parents, an Italian association. It's called ProRat Research. They really believe that the research is the solution. They are funding us quite a lot, and I hope they will be present at the next meeting together with you.